Hello, my name is Brandon Orellana, and today our learning objective is pretty clear. First, we'll go over four main differences between gene expression in prokaryotes like bacteria and eukaryotes, which are more complex cells. Then, we'll step back and look how gene expressions varies across different domains of life. By the end, we should have a solid understanding of not only what differences are, but also why they're important in biology.
Hi, I'm Lola, and I'm going to be going over gene expression. But first, what is gene expression? It's going to be the process by which a gene's information is able to create a protein. And there are two steps that a cell will go through in order to express the information from the gene, and that's going to be transcription and translation. Transcription is going to be the first step, which is the use of the enzyme RNA polymerase, which will synthesize a strand of DNA, and the strand of DNA will be used as a template in order to make the RNA. A strand of RNA begins to transcribe at the presence of a promoter, which is the sequence of nucleotides that codes for the start of transcription. In a similar fashion, there's also a sequence of nucleotides that codes for stopping transcription called a terminator. And by the end of transcription, a strand of RNA will be complementary and antiparallel to the DNA strand that it was transcribed from. In prokaryotes, mRNA, which is going to be the messenger RNA, which I'll get to later, can carry information from one or multiple genes, and the single gene carrier is going to be called monocistronic, and the multi-gene carrier is going to be called polycistronic. And moving on to the second part of gene expression, that's going to be translation, which is going to basically decode the strand of mRNA, and there's going to be three main structures used in this process. The first is going to be mRNA, as I previously mentioned, our messenger RNA. It's the first structure used and is a temporary replication of a strand of DNA, and it is made up of nucleotides, which code for a group of proteins. And each set of three nucleotides found in mRNA is going to be called a codon, and it's going to code for a specific amino acid. tRNA is going to be our second structure. It's what's going to carry what's called an anticodon, and it's going to correspond with the codon of the mRNA. And once that anticodon is delivered, the tRNA is then recycled in a process called charging, where the cytoplasm recognizes the tRNA and will attach the amino acid it requires. And the final structure used during translation is going to be a ribosome, which is going to align the amino acids and catalyze the formation of peptide bonds that hold them together. The process of translation as a whole is going to happen in three steps. Initiation is the first step, where the ribosome unit will attach to the start codon, which is AUG. Elongation is the second step, and it occurs when a full-length polypeptide is formed as each codon is paired with its complementary anticodon. And the final step is termination, where the stop codon is going to appear and the strand has officially been finished.
Hi, my name is Brandon Torzala, and I'm going to be talking about transcription in prokaryotes versus transcription in eukaryotes. So first, in prokaryotes, due to their lack of a nucleus, their transcription occurs within the cytoplasm instead of a nucleus, meaning that their transcription and their translation can occur at their same time. And there's only one type of RNA polymerase transcribing all types of the cells, such as mRNA, tRNA, and RNA, to help recognize the promoter regions, meaning that all three of those RNAs are only made by the one type of RNA. And in prokaryotes, they use the promoters called sigma factors that begin upstream on the gene at the -10 region, helping to unwind the DNA, and at the -35 region for their RNA polymerase to easily bind and initiate their transcription. In the first process, called initiation, the RNA polymerase sigma factor binds to the -10, -35 region, and the DNA unwinds, sigma factor is released, and that RNA chain begins. In elongation, an RNA polymerase makes RNA from the 5' end to the 3' end. This uses DNA as its template, taking inspiration from the 3' end and the 5' end in order to copy it. This then relates to termination, in which the RNA polymerase encounters the terminator sequence, This allows it to know when it needs to detach from the DNA and begin to release its new RNA or synthesized RNA copy. This mRNA is often polycistronic, meaning that it can transcribe for multiple proteins, not just one. So that's where it gets the one RNA polymerase type can translate for all three of the RNA. Versus in a eukaryote, it's due to a presence of a nucleus, their transcription occurs in the nucleus, but it also can be transferred into the cytoplasm, but that is for translation once transcription is over with. And since in eukaryotes they're monocistronic, that means that there is not one RNA or polymerase making all three of them. Each one has its own to make one. So an RNA polymerase 1 helps with rRNA, RNase polymerase 2 helps with mRNA, and RNA polymerase 3 makes tRNA. So each one has its own separate function and goal. And in eukaryotes, they use the promoter called transcription factor, such as a GTF, starting at the -5 region, also called the TATA box, and help the RNA polymerase 2 form what's called the pre-initiation complex and begin to bind and initiate our transcription. And in the initiation sequence, the RNA polymerase 2 requires transcription factors to begin, like the GTF, and they bind to that -25 region. In the elongation, the RNA polymerase 2 creates the pre-mRNA, which is before mRNA is created. And that includes the introns, which is then removed by the splicing when making the messenger RNA during elongation. And after it is spliced, the mRNA is made by capping the 5' end and creating a poly A tail at the 3' end. And during this termination of eukaryotes, after the cleavage at the poly A site has been capped by that poly A tail at the 3' end, it then leaves the nucleus to that cytoplasm after transcription is finished to begin translation. And that mRNA is monocistronic as it begins translation, meaning back to the three different RNA polymerases, the RNA polymerase 1, 2, and 3, which all have their own separate goals and their own separate functions within transcription. Thank you.
So in this slide, we have RNA processing in eukaryotes. There's 3 steps. So the first step is the capping, where a modified guanine nucleotide is added to the 5' end of the pre-mRNA. This helps protect the mRNA from degrading and helps the ribosomes recognize the mRNA during this translation process. Second step is about adding the poly A tail, and this is just stabilizing the mRNA molecule, aiding in the export to from the nucleus, and makes translation more efficient. And then third step is splicing, which is the introns from the non-coding regions getting removed while the exons in the coding regions are getting joined together. This converts the pre-mRNA into a continuous coding sequence and allows for alternative splicing, which is the process of different combinations of exons creating different proteins from the same gene. And the result is the pre-mRNA is transformed into a mature mRNA.
Hi, I'm Bryn, and my first slide is on introns and exons and the difference between them. So introns are the segments within a gene that are removed during RNA splicing of pre-mRNA, messenger RNA. They don't directly code for proteins, but they do have important roles in regulating gene expression, alternative splicing, and producing non-coding RNAs. Introns stay in the nucleus and are cut out during the splicing process. Exons, on the other hand, are the portions of eukaryotic genes that remain in the final mature mRNA. So unlike introns, exons do code for amino acids, and they are sequences that are actually expressed. So during splicing, the exons are joined together, forming the coding sequence that will be translated into a protein. So to sum it up, introns are non-coding sequences that are removed, while exons are coding sequences that stay and ultimately determine the product of the protein. Next is translation differences in prokaryotic and eukaryotic cells. So translation is the process of decoding the information that is carried in messenger RNA to synthesize the specified protein. In eukaryotes, translation begins after messenger RNA is produced in the nucleus. mRNA is first made as pre-mRNA, and this pre-mRNA must be processed to form mature mRNA, so that's a difference between eukaryotic and prokaryotic. Eukaryotic is first pre-mRNA, and prokaryotic doesn't have to be processed before it is just mature mRNA. And so during this process of maturing the mRNA, a protective cap is added to the 5' end of the DNA strand, and once the mRNA matures, it leaves the nucleus and enters the cytoplasm where translation occurs. Translation is divided into 3 main phases. The first is initiation, the second is elongation, and the third is termination. Initiation in prokaryotes begins when the 2 ribosomal subunits come together at the ribosome binding site on the messenger RNA. During elongation, the ribosome creates a full-length polypeptide chain through several steps. First, initiating transfer RNA occupies the P site of the ribosome. Next, a new transfer RNA carrying an amino acid enters the unoccupied A site of the ribosome, and then the ribosome can then catalyze the joining of the amino acids, transferring the growing chain from the tRNA in the P site to the one on the A site. The ribosome shifts forward by 1 codon in the 5' to 3' direction, and that's the translocation process. This causes the uncharged transferred RNA in the P site to move into the E site where it exits and then can be recycled and used again. This elongation cycle continues until the ribosome reaches the end of the messenger RNA molecule. And finally, during termination, translation ends when the ribosome encounters a stop codon, and special enzymes then release the completed polypeptide chain, and the ribosome detaches from the mRNA. And at this point, the ribosome can be reused to translate another mRNA molecule, so that's the recycling. It's important to know that the ribosomes act as a decoding site for mRNA and function as a highly efficient protein synthesizing machine. And then there's many factors that are involved in the translation of bacteria. So the anticodon, the mRNA correct reading frame, the ribosome and its binding site, ribosomal RNA or rRNA, the start and stop codons, and then transfer RNA as well.
Hi, I'm Brooklyn Trice. I'm going to be talking about gene organization and ribosomal structure. To begin, gene organization is the arrangement of genes within the DNA of a cell. First, we're going to start with prokaryote gene organization, which includes genes that are separated by non-coding DNA. Each gene has its own promoter. A promoter is a starter signal. Regulatory sequences, which allow fine control over gene expression. Most important part about gene organization in eukaryotes is that they are monocistronic, which means that each mRNA molecule codes for only one protein, and that enables specific regulation for every gene. Prokaryotes, their structure is composed of genes that are often grouped in clusters of genes with similar functions. There is only one promoter for several genes in prokaryotes. Also, most important for the organization, prokaryotes, they are polycistronic, meaning that each mRNA molecule can code for multiple proteins, and this allows groups of proteins with related functions to be produced at the same time. At the bottom is a little chart with the differences between the two. To summarize, eukaryotes are single gene and separated. Prokaryotes are genes that are in clusters. Eukaryotes are monocistronic. Prokaryotes are polycistronic. Eukaryotes contain only one per mRNA, while prokaryotes contain several per mRNA. Eukaryotes are highly specific, and prokaryotes are grouped and coordinated. Now on to ribosome structure. Beginning with structural features of prokaryotic ribosomes. Prokaryotic ribosomes, their size is 70, composed of large and small subunits. Large subunits are 50 and smaller ones are 30. rRNA content in prokaryotic ribosomes are 23 and 5 and 50 subunits, and 16 and 30. Protein content, there are 34 proteins in the large subunits and 21 proteins in the smaller subunits. Prokaryotic ribosomes are always free in the cytoplasm, meaning that they contain no organelles. The functional significance of prokaryotic ribosomes are that they are simple and efficient for rapid protein synthesis, as well as that they allow for coupled translation and transcription. Structural features of eukaryotic ribosomes. Eukaryotic ribosomes are composed of 80 ribosomes. Large subunits are 60, small subunits are 40. The rRNA content is 28, 5.8, and 5 in 60 subunits, and 18 and 40 subunit. Protein content is 49 proteins in the larger subunits and 33 proteins in the smaller. Eukaryotic ribosomes are free in the cytoplasm and attached to the rough endoplasmic reticulum. And then for eukaryotic ribosomes, their functional significance is that they are more complex structure for regulated protein synthesis, and they support compartmentalization in the nucleus and translation in the cytoplasm. Key differences between prokaryotic and eukaryotic ribosomes are their size. Prokaryotes are 70, while eukaryotes are 80. Eukaryotic ribosomes can be attached to the rough endoplasmic reticulum, while prokaryotic ribosomes cannot. And then gene expression is also different between the two. In prokaryotes, it is faster in simultaneous transcription and translation, while in eukaryotes, the gene expression is includes separate processes which allow for more regulation.
Hello, my name is Brandon Orellana, and I will talk about Christian worldview and science. To begin with, Christians have the belief that God is the creator of the universe, and that He designed this universe with order and purpose. Then, human dignity. Every person has value and worth because all humans are made in the image of God, as it is established in Genesis 1:27. Then we have to talk about truth and morality. Truth is not something we create. Truth comes from God. It is defined from His nature and teachings. Right and wrong are absolute and not relative. Christians often have the belief that the purpose of life is to build a relationship with God and others. Jesus is the only and ultimate path to truth, as it is established in John 14:6. Then science and religion. People often have the belief that science and religion do not go hand in hand, but the Bible does never contradict science. It provides a foundation for us to explore creation. Science explains how the world works and the details behind creation. Meanwhile, religion is the explanation behind the existence of the world and why every life matters.